Alright, so our next unit is going to be called Mendelian genetics. And the first thing we're going to do is go over some really important terms that go with the whole idea of genetics. The first one is traits. Then you have heredity and genetics. So trait is any characteristic that can be passed on from a parent to their offspring. So for example, in humans, you may notice like you have a similar nose to one of your parents, or your eyes are the same color as one of your parents. And those are traits like eye color, the shape of your earlobe, all traits that can be passed from parent to offspring. And the heredity component of that is the actual passing of the traits. And then genetics is the study of heredity, so the passing on of traits from parent to offspring. Hopefully some of this is review from last year. So the most important scientist to genetics, because he's sort of the father of genetics, the person who founded the actual study, is Gregor Mendel, and he was a monk. An Austrian monk, to be more specific, and he studied everything with pea plants. And you might be thinking, why would he pick pea plants? And that is something that we are going to discuss a little bit later in the video. And he developed what we now call the laws of inheritance. And there's another beautiful picture of Gregor Mendel. So he, over his studies, cultivated and tested 28,000 pea plants. Now, if you think about what makes a good experiment is that you can repeat it over and over again, and you have lots of different plants to test it on. So his experiments were good because he tested it so many times on a ton of plants and got the same results. So that's what made Gregor Mendel's contribution to genetics so important. It was because it was a really good experiment and no one had ever really thought of it that way before. So this is a lovely picture of Gregor Mendel's experimental garden. It is in the Czech Republic, if any of you feel like traveling there to see it. And it's a pretty cool thing. Alright, so we are going to be talking about the monohybrid cross and the dihybrid cross. Monohybrid is just that single trait. If you remember from 6th grade, you did do very simple Punnett squares, where all you did was deal with that one thing, like flower color, eye color, earlobes. And then dihybrid is 2 traits. So the example is flower color and plant height would be 2 traits that you could look at being passed down. And instead of a nice little box with 4 different squares in it, you have 16 different squares to fill in. But it's not that different of a process to the 4-square one. And those squares are called Punnett squares. And they are what you're going to be using to solve all of the genetic problems that will come up in class. And there is a lovely Punnett square. So basically each gene or allele is going to be represented by a letter, and you take that letter and you drop it down to the box. And as you can see in this picture right here, like this lowercase p goes to this box, and then this p goes to this box. You would put the uppercase P in this box and the lowercase p in this box. Uppercase P, uppercase P, uppercase P, lowercase p. And the very important vocabulary word is one that I already mentioned. I said the word alleles. And alleles are different forms of a gene. So you can have a dominant gene and a recessive gene. They both code for the same trait, like eye color, but you have 2 different forms of it. Alright, dominant genes are the ones that are going to be expressed over the recessive gene. Like if you have a capital R and a little r, you are going to have the characteristic represented by that capital R. It dominates the other gene and is the one that is expressed. Then you have the recessive trait. And in order to show the recessive trait, you actually have to have two lowercase letters. Because if you have that uppercase letter, once again, it's going to dominate, and that is what will be expressed. So if you have recessive genes, you have to have two alleles that are both lowercase. And that usually, statistically, recessive genes can occur less often in the ratios that we're going to look at, but in the population as a whole, sometimes having that recessive gene is incredibly important to your survival, which we will talk about in an article that I will give you to read. Another few words that you need to know are genotype. Genotype is the combination of genes, so that's going to be your letters. So you'll have capital letters and lowercase letters. So if you have a big R and a little r, that's a genotype, or a little r, or a big R, those are going to be what we're referring to as genotypes, because it's actually talking about the genes, whereas phenotypes is what it looks like. So if you have big R, it's going to be red. Red is the phenotype because that is what it looks like. So the example that is going throughout this PowerPoint is colors of flowers. So big R is a red flower, little r is a yellow flower. And genes occur in pairs. So you're always going to have two alleles that affect that specific characteristic. So with these two alleles, big R, little r, you can have a red plant represented by a big R, or a red plant with a big R, little r, because the capital R is the dominant gene and it's going to be the one that's expressed. And then you have little r, which will give you the yellow phenotype. Alright, your genotypes have special names for them because in science we're all about using the big words. Homozygous is a genotype, which means that both letters are the same. So you're either going to have two capitals or two lowercase. And they are the same, hence the word homozygous, homo meaning same. So these are called pure because they carry the same allele for the gene in both instances. So you'll have two dominant or two recessive genes. Then you have heterozygous. Hetero, the Greek root for that is different. So you're going to have a capital R and a little r. So you'll have both dominant and recessive. That is heterozygous. And those are referred to as hybrid. And now we're going to get back to that question is, with all of these new vocabulary words, why did Gregor Mendel use pea plants? What made them so special? Well, for starters, he could grow them in a small area. He didn't need a vast amount of land to grow these pea plants. He could put it in a nice little garden. Then, the pea plants produce lots of offspring. And that's really good because then you can have different genetic variation based on the offspring. And then he could also self-pollinate. So these plants would self-pollinate and they would create the same color of flower. So they were pure. So if you had a white pea plant and all it ever did was make white pea plants from generation to generation, you would know that that was a pure genetic, it would have a pure genotype. Also, pea plants can be artificially pollinated, so you can take pollen from one flower and transfer it to another. And if you remember from class, I've said a couple of times, pollen is plant sperm. So you're taking plant sperm from one flower, putting it on the female part, the pistil of the flower, and crossing them that way. In a lot of experiments, they put like bees on sticks, and they use that to transfer the pollen from one plant to the next. So, pollen contains sperm. Pollen is plant sperm, and that is found in the stamen of the flower. If you look at this picture, you can see the stamen, and then there's the pistil and the ovary, which the eggs are found in. And we will talk about that a lot more when we get to when we start studying plants in the spring. So, self-fertilization is when the sperm that's made in the stamen of the flower pollinates the the egg in the pistil of the flower, and so it just makes more of itself, whereas cross-fertilization happens between two separate flowers, and they get the sperm from a different flower. So basically, Mendel hand-pollinated these flowers by using a paintbrush, which would work by collecting the pollen and then sticking it on another flower's pistil. So he snipped the stamens so that they wouldn't self-pollinate, so then that wouldn't be a gene that was factored into his experiment. And then he covered each flower with a cloth bag. And he traced the traits through several generations. So he would have like a parent generation where he crossed these first two plants, and then he would look at those offspring, he'd cross them, and then see what he got. So he could see all of these generations of pea plants. So the pea plant's traits that he looked at were seed shape, seed color, pod shape, pod color, seed coat color, flower position, plant height, and flower color. These were what he was looking at, and he looked at each of them separately, really. So he would take the pea plants and create plants that only produced round seeds, and then plants that only produced wrinkled seeds, and then he'd cross them and see what the results were in the next generation of pea plants. He did the same with all of them. And as you can see, he has different letters that he used. You have R, Y, S, G, depending on what the trait was. So here you can see seed shape, you have smooth, wrinkled, yellow, green, pod shape, inflated, pod shape, wrinkled, pod color, it'd either be green or yellow, and you'd have purple or white, or the position would be on the stem or at the tip, depending on which plants he was going for. And then you have tall plants and dwarf plants. So basically, he performed all of these experiments and he got these results. And you can see that the dominant trait was the smooth seed. So he got 5,000 smooth seeds. The recessive trait was the wrinkled, and he only had about 1,850 of those. However, I do want you to keep in mind that Mendel's experiment worked really well for pea plants, but when you look at human genetics, it's much more complex. Much more complex than these pea plants. And once again, there's an article that you'll read that will explain that a little bit better. So the average ratio from dominant to recessive was a 3-to-1. And you can prove that in Punnett squares, which there is a diagram in your book that shows that. So theoretically, if we cross two pure plants, we are going to get 3 round, 1 wrinkled. The larger sample that you have, the more pea plants you cross, the more results you get, the better your ratio is. But you would have to do thousands upon thousands of pea plant experiments to do that. And he would start with the parental generation, which is abbreviated P1, and that would go to the F1 generation, or the first generation offspring. And then you have the F2, which is the second generation offspring. And we are going to be referring to that in the questions as what is the F1 generation, what is the F2 generation, and you really need to know what those two terms mean. It's first generation and second generation. So for example, you have the P generation, you cross it, you get that F1 generation, and then you have the F2 generation. If you notice, you have 2 small, and then you have 2 tall, and then you have 3 tall and 1 short. That's a 3-to-1 ratio, which is what Mendel found in his experiments. And that is pretty much where we're going to leave off for the Intro to Genetics. We're going to go a little bit more into the monohybrid crosses next, as you can see on the screen, but that will be in the next video.